Imagine a tiny frozen world, hundreds of millions of miles from Earth. It should just be a dead, boring ice ball, but it's not. Today, we're going to dive into the incredible scientific detective story that's unfolding on one of Saturn's moons, Enceladus.
So this is the big one, the question that drives this whole hunt. Could this little icy moon of Saturn actually hold life? I know, it sounds like science fiction, right? But as you're about to see, the evidence is pointing to a really tantalizing possibility. This could be one of the most promising places to find life beyond Earth.
So let's meet our main suspect, Enceladus. This place is small, only about as wide as the United Kingdom. And if you just looked at it, you'd think, yeah, that's a quiet frozen wasteland. But you'd be wrong. Underneath that icy shell, something absolutely extraordinary is going on. And this is where the mystery really kicks off. NASA's Cassini spacecraft flew by and saw this. Giant plumes of water ice and gas just erupting from these huge cracks near its south pole. This tiny moon was alive, geologically active, and literally shooting its own ocean out into space. And just like that, the hunt was on. Okay, so to figure out if life is even possible here, we need a kind of framework. Astrobiologists have a checklist. For life as we know it, you need 3 key things. You need liquid water, you need a source of energy, and you need the right chemical nutrients, basically food. So, does Enceladus have what it takes? Let's check the evidence. First up, we're investigating the scene of the crime, so to speak. We've already confirmed that first ingredient. There's for sure a liquid water ocean under all that ice. But just having water isn't enough. Is it a place where life could actually thrive? The really critical question is there an energy source? And those plumes gave us our first major break in the case. Cassini flew right through them, and it found way more than just water. It found tiny little grains of silica, molecular hydrogen, and even some complex organic molecules. Now the silica and the hydrogen together, that's the smoking gun. It practically screams hydrothermal vents on the seafloor, places where hot, mineral-rich water is cooking up some interesting chemistry with the rock. To really wrap our heads around what this means, scientists look at similar spots right here on Earth. Deep sea vents, sometimes called white smokers, are just teeming with life, and none of it relies on sunlight. By studying hundreds of these places, we can build a pretty good model for what's happening on the ocean floor of Enceladus. And this brings us to a key process with a fancy name, serpentinization. But all you really need to know is this. When seawater mixes with certain kinds of rock, it makes molecular hydrogen. And for any little microbes that aren't using sunlight, hydrogen is like a 5-course meal. It is a potent source of chemical energy. It could be the engine powering a whole deep-sea ecosystem. So exactly how much energy are we talking about? Well, a recent study modeled all of this using what we've learned from Earth. And the number they came up with is, well, it's absolutely staggering. It completely changes the game. Get this, the study suggests Enceladus might have at least 100 times more chemical energy available for life than we ever thought. 100 times. So this environment isn't just habitable, it could be incredibly rich, just brimming with the energy life needs to get going. Okay, so we've established a prime location for life. The scene is set. But that's not enough, right? That's just circumstantial. Now we need to find the direct evidence, the actual chemical fingerprints that life would leave behind. You know, if life exists, or ever existed, in that ocean, it's got to leave behind some specific molecular clues. We're talking about the fundamental building blocks here, little fragments of DNA, lipids from the walls of cells, and all the chemical leftovers from life just doing its thing. Finding these, that would be unambiguous proof. But think about this for a second. How in the world do you detect these tiny molecules inside a tiny ice grain that's flying past you at thousands of miles per hour, millions of miles from home? Yeah, you're going to need a very special forensic toolkit for that. And this is the incredible solution they came up with. You can't really build a gun that shoots ice grains that fast in a lab. So they did something even smarter. They did the opposite. They fire a super fast laser at a tiny little jet of water. This creates a tiny explosion that perfectly mimics a high-speed impact. It lets them see exactly what a detector on a spacecraft would see. The process itself is just brilliant. Here's how it goes. The laser blast instantly vaporizes the water, making a cloud of charged particles. Then, an electric field makes them all race down a track. The lighter particles are faster, the heavy ones are slower.
At the finish line, a detector records who arrives and when, creating this graph called a mass spectrum, a unique chemical fingerprint of whatever was in the water. So, now for the moment of truth. We have the method. We know what we're looking for. By putting actual biological material into this machine, we can finally see what the smoking gun signature of life would look like to a spacecraft. Researchers took components from real bacteria, stuff like E. coli and a marine bacterium, dissolved them in water to make a kind of life soup, and ran them through the experiment. So, what did they find? And here it is. This is the mass spectrum for DNA. Each one of those labeled peaks corresponds to a specific part of DNA, like adenine and cytosine, the actual letters of the genetic code. You can see them right there. The pattern is unmistakable. This is what the fingerprint of DNA looks like. Now check this out. This is the signature for lipids, the molecules that make up cell walls. You get this very distinct pattern of fatty acids. See, life tends to be efficient. It likes to build these molecules 2 carbon atoms at a time, so you see a lot of even-numbered chains, like C16 and C18, and that's exactly what we see here. It's a powerful hallmark of biology. Of course, you might be wondering, but wait, Enceladus's ocean is salty. Won't that mess with the signal? And that's a great question. The researchers tested exactly that. They added salt to the water, and guess what? The key biosignatures, especially from DNA and lipids, were still clearly detectable. The method works. So, where does all of this leave our investigation? We've connected the dots, from a mysterious plume all the way to a potentially life-sustaining ocean. And we've developed the exact forensic tools we need to find the evidence. So, let's recap the case file. We have a prime location with a massive amount of chemical energy, way more than we thought. We have a proven forensic technique to analyze its ocean from space. And crucially, we now have the reference fingerprints. We know exactly what the signature of DNA and cell membranes would look like to a detector on a future mission. You know, we are on the verge of being able to answer one of humanity's oldest, biggest questions. The tools are ready. We know what to look for. All that's left is to go back and take a look. And that leaves us with one final, profound question. If a future mission flies through those plumes and sees these exact signatures, what happens next?